And first speaker is Professor Jianping Si from National University of Singapore. He received his bachelor's degree from Tsinghua University and PhD from Singapore SMA program. And then he joined NUS in 2010. And he is also associate editor of Aggregate. His title of the talk is Total Synthesis of Metallic Molecules. Please join to welcome Professor Jianping Si.
Okay, thank you very much. Very good morning. Thank you very much, Professor Yoon, and for the kind invitations. And it's my great pleasure and honor to be here. And let me share my slides here. So can you see my slides?
Yes.
Oh, yeah. Okay. So I'm very excited to be here, especially I'm the associate editor of the Aggregates. And today I will discuss our molecules. We call them metallic molecules. And my discussion a little bit, different from the other speakers. And of course, they were focusing on bioimaging applications. And my discussion is more on the chemistry part. So it's a materials. So hopefully, and this can enhance some of the collaborations and I can convince you. So this is a very promising materials that can be used for the bioimaging applications and also for other biomedical applications. Okay. So my name is Jianping Xie. I'm from National University of Singapore. And this is the title of my talk is a total synthesis of metallic molecules. And this is my molecules. And you can see here, the orange here is the metal actants like gold and silver. And the yellow is like the organic ligands. So it's like the sulfur actants. So these molecules, it's very small. It's about 1 nanometer. And we can also know the molecular formula, right? So we know the molecular formula, like we can know how many metal actants inside and how many ligands on the surface of these molecules. And as a functional molecules, in these metallic molecules, they have very unique physical, chemical, and biological properties. So one of the properties I will discuss today is their optical properties, like the fluorescence properties. So if you want to realize the very consistent and high performance, and we may need to produce a very high quality of these particular molecules. Okay? So today my talk will be focused on the first one. So we call the precise synthetic chemistry. So the meaning of these metallic molecules is at the atomic level, we want to control the size, composition, and surface and structure of these particular molecules. So if we want to develop a precise synthetic chemistry, so we need to understand, so how does this form, right? So this will be, the focus of my talk will be the total synthesis. So we want to unravel the reaction pathway of these particular metallic molecules. So actually the total synthesis of metallic molecules is got the inspiration from the organic chemistry. So similar as Professor Yoon and Professor Tang, right? So you know, organic chemistry is very powerful. So you can easily use 4 elements like hydrogen, carbon, nitrogen, oxygen, and then to create unlimited molecules like the small drugs, okay? Or complicated polymers, or more complicated like biomolecules like DNA and proteins. But if you check our periodical table, so majority elements are metals, but for these metals, they are mostly crystalline structure. So they are really lacking of what we call a molecular-like structure diversity. So the questions or our interest is, can we use these metallic elements, like very common elements like gold and silver, and then to use to create a universe of these metallic molecules? So this molecule, metallic molecule, is similar as our organic molecules. So for example, they have molecular structure, they have molecular formula, and more importantly, they have some properties, molecular properties, right, that we can use for the different applications. So why we are interested in these particular molecules, or we call metal nanoclusters, and simply because they have very unique physical, chemical, and biological properties. So I just give you one example. So for the metal elements or materials, they have this we call size-dependent properties. So for the size-dependent properties, for example, they have this size-dependent optical properties. Okay, so we all know this, right? Nobel Prize is about gold, and this gold they have is in yellow, right? Yellow color. So that when we decrease the size of our materials, so for example, if we have thousands or even millions of atoms aggregate together, and we have very small particles, and we call them nanocrystals, and we can start to see the different gold solutions. Like we have a red gold solution, we have a blue gold solution, and we also have this black gold solutions. So this is because it's from the continuous electronic states, or they have very interesting surface plasma resonance properties. And we can control these properties by controlling the shape, size, and composition of these materials. Right? And if you still remember, well, our young students, right? If you still remember our high school chemistry, so you know we have for single gold atoms, it has the discrete electronic states. So the next questions we may ask is, so what will be the transition between a single gold atom, discrete electronic states, to these thousands of gold atoms, continuous electronic states? Right? So the transition is the materials I will discuss today, is aggregation of a number of these gold atoms. So these are nanoclusters or gold molecules. So they are very small, and typically they're less than 1 nanometer and less than 150 atoms. So they have this discrete electronic states. So different from their larger brother, like gold nanocrystals, they don't have these SPR properties, but they do have some of these we call molecular-like properties. Like now, gold, under the UV light, you can see they can show the different fluorescence. They also have some optical absorption and catalysis properties. So as a materials chemist, and we can control the size, composition, surface, and structure, and then to control the properties of these materials. Right? So for metal nanocluster, one of the properties we are using is the fluorescence properties, and they can use for the different applications. So for clusters, we use another 2 slides to give a brief introduction of the properties, the optical properties. They are very promising optical materials. Because for these particular molecules, if we can control the core, that will be a metal. So it can be gold, it can be silver, and the shell, right? So they're protected by organic molecules. So we can control the core and shell of these particular molecules, and we can then control the interaction of this metal nanocluster with light that have unique optical absorption like photoluminescence, and then we can use the luminescence property, for example, for the bioimaging application, biosensing applications. So another properties for metal, noble metal, is they are very good catalysts. So we can combine the catalysis properties and the optical properties, and then we can design like photocatalysis, right? And so that can be used for many applications as well. So we have started to work on luminescence properties of metal nanocluster more than 10 years ago. So it's very lucky we got the inspiration from Professor Benzhong Tang on the AIE properties. So we call it aggregation induced emissions in the organic molecules, right? So we found out for the inorganic molecules, like metal molecules, so they have similar these aggregation induced emissions. So one of these aggregation induced emissions is when the molecules, they aggregate together, and then the luminescence will be increased, right, enhanced. So when we dissociate these molecules, and then these molecules, the luminescence will decrease, right? So that's we can easily see from this video. So when we aggregate this is gold one, tight complexes. So when we aggregate these molecules, so they show a very strong luminescence. And when we dissociate these molecules, like we spray the water, so it can dissociate, and then luminescence will disappear. And we spray like external, so that's a back solvent. So they can induce the aggregation of these molecules, and then the luminescence come back, right? So this is give us information where our molecules have this aggregation-induced emissions, AIE properties. And then we can control the different level of these aggregations. For example, we can control these like gold-1 tight complexes on the surface of this gold nanocluster at a single cluster level. Or we can further aggregate our molecules, right, to form these supramolecules. And this is beyond single cluster level. And then we can control the properties of these clusters, or we can control the link, like the structures and the motif on the surface of this cluster, and then to control the emission color of this metanol cluster, like all the way from yellow, orange, red to near infrared, right? So the clusters. So these molecules, and they are very small, like they have show a very stable and bright luminescence. They have very good biocompatibility, like gold nanocluster. So they are very small. After they serve their function, they can be cleared by our body. So we call it renal clearance. And then our clusters are also protected by biomolecules. Like we can protect it by the peptides. So they have very rich surface chemistry. So they are very promising. We call luminescence probes for different imaging applications, and also for many biomedical applications. So today my talk will not focus on the bioimaging applications. So we will go to fundamental of these particular molecules, and hopefully my discussion that can convince you this is a very interesting type of materials that we can use for different applications. So we will be working on these molecules in the past 14 years. So we call metallic molecules, or they are the atomically precise metanol cluster. So I have different applications. But if you want to support all these applications, or you want to have very promising applications, consistent applications, so we may need to go deep, right, to understand these particular molecules. And we need to have a protocol that can help us to produce high quality of these molecules. So this is related to, we call cluster chemistry. So we have been working on in the past many years. So actually we are working on two topics. So first one is for material synthesis. So at the atomic level, we are trying to control the size, composition, surface, and structure of these metallic molecules. So the second one is the fundamental properties. Because for all these applications, so we look at these functions of these particular molecules. But some of the fundamental properties, like the optical absorption and cluster structure and luminescence, so they are not very well understood. So the second topic we are working on is at the molecular level. We are trying to understand the fundamental properties of these particular molecules. And this is a very brief introduction of my research in the field of this metanol cluster, from the synthesis, fundamental properties, and all the way to the applications. So today my talk will be just focused on the first part. So I will just give you one examples, and for the synthesis of these metallic molecules, or the questions we are trying to address. The fundamental questions may also be applied to other inorganic or even organic molecules. Okay, so we are focused on the cluster synthesis. So if we are working on inorganic nanostructure material synthesis, so you often have the same questions. We call it open questions in organic nanomaterial synthesis. So now I will just discuss in these slides. So we use this very common cluster, like gold 25. So there are 25 gold atoms and 18 tightly ligands. So the orange here is the gold atoms. So this brown is the sulfur atoms. So we can design a very simple method to synthesize these molecules. Like we have gold acids, we have protecting ligands, and we use a water. And after we add in a reducing agent, so it's in this reactor, so we can start to see the color change, and from yellow, and you can see from yellow, orange, and to brown, and finally to reddish brown, right? So the synthesis is very simple. So after 10 minutes, and you can open this reactor, and we can precisely obtain our final products. But the questions we may ask is, so what exactly happening inside this reactor? We see the color change, right? So like definitely something happening, okay? But this is a totally black box. So we know the input of this reactor, and these are our input. So we know the output, our final products, but we don't know what exactly happening inside this reactor. So this is our open questions for the precise synthetic current chemistry. So one key question is, so what are the key intermediates towards the formation of these final products? So can we identify all of these key intermediates, right? So if we can identify the key intermediates, so can we write down the step-by-step reactions? So along the synthesis of these particular final products, right? So this is similar as the total synthesis of the organic molecules, right? And if we can understand the key intermediates, we can understand, we can write down the step-by-step reactions. So the next, can we precisely control these molecules, right? Like composition, size, structure at the atomic level. But more importantly, at the predictable manner. So if we want to a particular clusters, then we can design a particular like protocol to synthesize this cluster. So finally, the question is, can we achieve total synthesis of these metal nanoclusters? Right? So this is a very interesting and very challenging questions. And actually, for the total synthesis, it got inspiration from organic chemistry. So they're very powerful concept, right? So I just give you one example. Like this is the quinine. It's a very important drug. It's a complicated molecules. But in organic chemistry, we can use a very simple precursors. And then we can design more than 20 steps to precisely synthesize these particular molecules. And we can also know each of these steps, right? So the questions we ask for the metal nanocluster synthesis is, so can we achieve the same? So can we identify all these intermediates and then understand the steps involved in the synthesis of these clusters, right? So the open question, if you go back to the previous slides, so we know is, so we need to have some of the methodology that can, or platform to help us to monitor or to identify the key intermediates towards the formation of final products. So my group will propose to use this very simple, we call mass spectrometry. So it's a real-time mass spectrometry. So if we synthesize our cluster, so we can directly fuse our reaction solution inside the ESMS, the mass spectrometry. And then we can identify all the key intermediates. You can review reaction kinetics and dynamics at the atomic level. And if we have some understanding, we can unravel the reaction pathway for all species. So actually our platform is very simple. I can just demonstrate in this the video. So this is the synthesis of a very common cluster, the Go25 SR18 clusters. So you can use the ESMS to monitor the synthesis of these clusters. So as shown here, so before the reactions, so you can see some of the peaks here. So when you add in a reducing agent, so we will start to see some of the peak disappear and some of the peak related to products that continue increase, right? So if you can analyze this mass spectral, and then we can watch the formation process. And now we have the molecular formula informations, right? And we can analyze this spectral, and then we can review the reaction chemistry at the atomic level. So for example, for Go25 SR18 nanocluster synthesis, we can identify all these intermediates. And we can also know the time-dependent abundance of all these intermediates. So from the analysis of this, like for if two intermediates, the abundance decrease and one increase. So there's some correlation between these three species, right? And then we can write down like step-by-step reaction towards the formation of these final products. And we can also identify the different reactions by using this very simple method like reduction reaction, addition reaction, right? Okay, so we will summarize our study. I mean, in the past like 10 years, and in this particular field, in this slides. So we call a precise synthetic chemistry. So we have coined our concept and in accounts 2018. So we call it total synthesis of metallic molecules. Okay. So we use a very simple platform like the mass spectrometry together with the tandem mass and also the optical properties, the UV-based absorption spectroscopy. And but important for this particular research direction is to ask questions. Right? So what are the fundamental questions we are trying to ask for the synthesis of these particular molecules, like the metal nanoclusters? So I will just summarize in these slides. So it's like for clusters, like Go25, SR18 nanoclusters, so we can use a very simple method to synthesize this cluster. But the questions we may ask is, so how will these gold ions reduce to form the gold atoms? So how will these gold atoms aggregate together to form a small clusters? And how will these small cluster continue growth to form a large clusters? So these are open questions we call reduction growth. And why the final product is AU25, not AU24 or AU26, right? So we use our platform trying to understand this reduction growth. So we can go further, similar as organic molecules. Like we can use this cluster, small cluster, as a seed, and then we can further grow a larger cluster, like this AU44. So we are trying to understand the seed-mediated growth, right? So we can go further, and then we need to like form a very large crystals. So then from this line, we are trying to understand from the precursors, small cluster, large cluster, finally to a large, the gold nanocrystals. Right? So similar as organic molecules. So if we synthesize a particular molecules, you want to diversify or these like properties of these molecules, then we can modify, right? Or functionalize these molecules. So we can do the same for metal nanoclusters. So for example, if we want to adjust the properties of this cluster, so one very simple is we can do alloy, right? So we can use a second metal to dope inside our clusters. So for example, you have gold nanocluster, so we dope one silver atoms on the cluster. But the questions you may ask is, so whether we dope in this position or in the center. So we need to understand these alloy processes, and our platform can help us to understand this we call the precise alloy chemistry. So similar as the core of these clusters. So the equally important for a cluster, especially for biomedical applications, is the surface, the ligand of this cluster. For example, if you want to use this cluster to target the cancer cells, so then you may need to replace some of the ligands, right? So, and that can help you to target the cancer cells. And this can be done through a ligand exchange. So the questions may ask is, so what would be this particular process? So can we precisely control the second ligands, like in this position or this position? So if you have two types of ligands on our cluster surface, so can we control the number of these ligands and also the landscape of these ligands, right? So this is related to the surface chemistry. So similar as organic molecules, we can also form the supramolecules and by using the cluster as the building block. So we can do the several assembly and then to form the supercrystals. So the questions asked is, so what will be these several assembly processes, right? And then this is related to metal materials. So these are the summary of our research in the past 10 years and in the field of this called total synthesis of metallic molecules. So I will just use this as an example. So this is my last slide, is using the cluster. If you can derive the different chemistry, so you can use the AU25 SR18 as a precursor. So we have isomerization reaction, ligand addition reaction, several assembly, alloy reaction, and then we can divert it to or synthesize other functional molecules, right? So this is similar as the organic molecules. And recently, we also developing we call a smart synthesis. So we integrate this AI, and then we can do a high throughput screening, and together with this automatic synthesis. So this is my last slide I will show you. So we can use, now we have developed this automatic synthesis, and that can help us to synthesize these particular molecules. So let me this. So these are the formation of the gold nanoclusters, and then we integrate this in situ characterization techniques to help us to synthesize this high quality of these molecules. Right? So let me just summarize my talk today. So we have discussed the total synthesis of these metal nanoclusters. So hopefully I can convince you this is a very promising materials that we can use for the different applications. So if we can precisely synthesize them. So I will end my discussion by using this slide to thank my group members, collaborators, and funding agencies. And most important in these slides, so it's welcome to Singapore and welcome to NUS. Thank you, Professor Lim. So you have questions? I'll be happy to take your questions.
Okay, thank you so much for your nice talk. Very interesting. I think the audience can upload or send a message with questions in the Q&A session. So if you would like to, I have one question in the Q&A, and is how solvent matters for the synthesis of AU25, and can we able to synthesize only pure AU25? Yeah, first question is.
Okay, so this is very interesting questions. Yes, so for these molecules, it's similar as organic molecules. So if you can design a very efficient synthetic protocol, so they can produce like very pure these particular molecules like AU25. So the rest will be some byproducts is complexes. So like no other clusters. Because this AU25 is the most stable, like the metallic molecules and in existence. Yeah.
I have one question for you. Actually, the ligand exchange surface decoration is very interesting. And have you tried it, or you are planning to try to like introduce the biomarkers on the surface, or?
Yeah, we have thank you, Professor Yoon. So we have tried to like integrate some, for example, like the peptides and on the surface of these clusters and just trying to precisely control this. Yeah, that's a very interesting directions. But for some complicated molecules, is a little bit challenging because they might not be compatible with the ligands on the surface of cluster. So we use a different method and trying to incorporate some of the biomolecules or bioactive molecules on the surface of the cluster.
Thanks. We have one more question. What are the advancements of nanoclusters in comparison with the nanomaterials?
This is a very interesting question. So like one of the advantages for this cluster is they're very small, right? So they're less than 2 nanometers. So after they serve their function, for example, for biomedical applications, so they can be completely cleared by the body. So for the large particles, they may accumulate inside the body. So this is one advantages. So the second one is, and for this cluster, it's more like the molecules. So we can precisely synthesize these molecules. We know their molecular formula. We know their molecular structure. So these are the difference from some of the large nanoparticles. Okay? So it may benefit some of the biomedical applications because we know exactly like these particular molecules.
Okay, thank you so much for your wonderful talk. Thanks to Professor Jianping, CEO, for his wonderful talk. Okay, thank you. Yeah.
Thank you, Professor.
Yep. Actually, I forgot to introduce myself. This is Juyoung Yoon from Ewha Women's University, and I am also working as editorial board member of Aggregate. Professor Benja Tang actually joined a little bit late, so who is the chief editor of Aggregate from Chinese University of Hong Kong in Shenzhen. He will give actually the opening remark.
Okay. Yeah, good morning. Can you see my screen?
Yes.
Okay. Yeah. Thank you, Professor Yoon, for your kind introduction. And also thank you for organizing this webinar. I'm sorry I had a difficulty in entering the Zoom, and now finally solved the problem. Yeah, Professor Xie has just given a wonderful talk about the metal nanoclusters. And so now let me introduce our journal to you, because this is the webinar, the global webinar for our journal Aggregate. So first of all, let's see what is Aggregate. People are talking about the molecules. Okay, as you can see from here, so far most research are concentrated on molecules. However, seldomly we use single molecule species in our real applications. Okay, so when the molecules are aggregated, you get an aggregate. So the example here, like the nickel crystal, one single molecule will not have nickel crystallinity, but when many, this kind of mesogenic molecules are aggregated together, you get nickel crystals. Okay, and for aggregation-induced emission, single molecule species does not emit, but when they form aggregate, they will emit very efficiently. There's many such examples. Okay, so in other sense, probably it's the time to think about the paradigm shift from small, tiny microscopic molecules to meso or even micro, very big, aggregates. So what is the aggregation? Aggregation is a process or gate through which molecules assemble into complex hierarchical structures at the nano, micro, meso, or macro scales. So aggregate science has a very broad scope. First of all, because it's very broad in scope, so the area is multidisciplinary. Okay, it involves chemistry, material science, physics, biology, medicine, engineering, and so on and so forth. And in terms of aggregates, you can have many different things. Okay, dimer, oligomer, polymer, dots, gels, clusters. Professor has just talked about the gold nanoclusters. Okay, you can also move to anode, hybrid, crystals, film, and in biological field, the proteins, DNAs, cells, pathogens, all kinds of things. Okay, of course, in materials field, you have frameworks like Morph, Corf, Hoff, and composite materials. Okay, so here Aggregate has published many papers. Okay, so from the size of the letter, you will know that many people are talking about polymers, material science, and clusters, many different topics. We have also published some special issues. Okay, one is on photovoltaics, another one is on nanoscience. And we also have a special issue focused on emerging investigators. Some special collections are in progress, including distinguished Australian researchers, aggregation disk processes and functions, condensed matters, and the special collection dedicated to Professor Ben Feringa, the Nobel laureate for molecular machines. So we have a very strong editorial team. Professor Anjun Qing is the deputy editor, and we have an editor from Australia, Japan, UK, Canada, and US, Singapore, and China. Okay, so these people are very young, very energetic, and also very active in their field. So the editorial team is international with diverse background And as I just mentioned, they are all active researchers. So our board, editorial board, is very big. Okay, it's about 130 people from China, Japan, Korea, Singapore, US, UK, Germany, France, Canada, Australia, Spain, Italy, India, etc. 42 of them are listed in the Aggregate 2023 highly cited researchers. So, the publication is growing very fast. In the first issue, we published 9 papers, and now, last year, we published 137. And this year, the number will become even bigger. So now, it's a bi-monthly journal. Every issue, we publish 10 to 40 articles. And, the number of publications per year is growing, as just mentioned here. And the review is very fast, okay? So we reached the first decision in about, in less than 2 weeks on average. And it's an open access journal. And, because it's open access, so people can freely, read your papers published in our journal. So this is a historical development. We published our first issue in the end of 2020. And in less than half a year, it's included in this DOAJ. And in 2021, okay, in about 9 months, it was included in ESCI. And in the middle of 2022, Aggregate published the first JCR report on our journal. And our journal was ranked in the Q1 category in chemistry multidisciplinary, material science multidisciplinary, and chemistry physical. And last year, in the middle of last year, we got, our first impact factor of 18.8. And, again, we were in the Q1 of 3 categories. And, in August of 2023, Aggregate is included in Scopus. And, the Chinese Academy of Science, okay, put our journal in the Q1 category of chemistry at the end of last year. And it is anticipated that, Aggregate will be included in SCI in this year. So, we have organized many international events. See, for example, we have this Aggregate Angwangtai Forum. Okay, it has run, several times, and it was well received. We have this global webinar, and now it's the 8th, okay, global webinar for Aggregate. And the last one was held in the US. The organizer was Professor Wei Tao from Harvard Medical School. And this time, of course, is organized by Professor Yoon. And there's links, okay, to the previous webinar. You can, click it, and you can watch the webinar of, webinars held in the previous years. We also have awards. The first Aggregate award was given to Professor Bin Liu of the National University of Singapore. And we also have an award to young scientists, to best posters, and best papers, and even this, best cover images. So in summary, the Aggregate is developing very fast. Our first impact factor was 18.8, and it was ranked in the Q1 category of 3, areas by Aggregate, and it's ranked in Q1 of chemistry by Chinese Academy of Sciences. and the scope is very broad, and the readership is multidisciplinary, and review is fair and fast, and the publication is timely, and it's an open access journal. So this is our editorial team, and these are the QR code to our journal, and Google Scholar we also have a QR code here. So today, thanks again to Professor Young. We have invited Professor Xie, Professor Xu, Professor Lim, and Professor Chen to deliver talks in their field. Okay, and I have just listened to Professor Xie's talk, and it was really, educative and informative. And I hope, our audience will enjoy the rest of the lectures. Thank you.
Thank you so much, Professor Bender Tang, for a very kind introduction. So we'll move to the next speaker, Professor Zhao Chao-Chi, and he got his PhD from Dalian University of Technology, and he did his first postdoc in Ewha University, and then second postdoc in University of Cambridge. And he moved to Dalian Institute of Chemical Physics, CAS, where he is currently a professor. His title of the talk is Aggregation Disaggregation Mechanism Regulates Fluorogenic Probes for Super Resolution Imaging. Please join me to welcome Professor Zhao Chao-Chi.
Thank you, Professor Young. Thank you. I would like to thank Professor Young for the kind introduction, and also for the kind invitation to give me a chance to talk about my research in this webinar. I'm so excited to hear Professor Young host my talk. Several years ago, I worked with Professor Young for 2 years in Korea. It's a very nice time. My name is Zhao Chao-Chi from Dalian Institute of Chemical Physics, CS, China. And I'm honored to present our recent work on fluorescence probes for super resolution fluorescence imaging. These fluorescence probes are increasingly being found to be related to the process of aggregation and the disaggregation of fluorophores. And what is shown here is the temporal and the spatial dynamics of mitochondrial membrane potential. What is come to fluorescent aggregates, the well-known phenomena are aggregation-caused quenching, ACQ, and aggregation-induced emission enhancement, ARE. It has been more than 20 years since ARE was discovered. Besides being a novel type of fluorophore, I believe the greater significance of ARE research lies in its ability to draw attention and provoke contemplation within the scientific community regarding molecular aggregates, just like Professor Tang talked. This shift moves away from the notion that molecules are the smallest functional units. Perhaps molecular aggregates are the true functional units. In the study of fluorescence probes for super resolution imaging, we are currently undergoing a shift in focus from individual fluorophores to fluorophore aggregates. And I'm delighted to take this opportunity to introduce how the aggregation and disaggregation of fluorophores serve as a key mechanism in controlling super resolution fluorescence imaging. Rhodamine is currently the most widely used fluorophore in super resolution imaging. Apart from its high brightness and excellent photostability, rhodamine's switchable response due to its spiral ring enable it to break the difference limit. Rhodamine itself is an ACQ fluorophore, but subsequent research has shown that its aggregation plays a crucial role in regulating the rhodamine spiral ring switch. This is particularly important in the process of identifying targets in super resolution imaging. My talk will begin with a brief introduction to the principles of super-resolution imaging to provide an understanding of the special characteristics required for fluorescent probes in super-resolution imaging. And we will then focus on rhodamine as the primary fluorophore to illustrate the main theme of my talk, the role of fluorophore aggregation and disaggregation in super-resolution imaging, the challenges that exist, and finally, the method we have developed to address these challenges. An ideal fluorescent probe should rapidly enter cells and selectively light the target molecules. For instance, what is shown here is a probe from my lab that can enter cells and recognize and then light the mitochondria and the cell nucleus within 100 seconds. But when imaging through a fluorescent microscope, the emitted light from a point source is diffracted upon passing through the objective lens, resulting in an image spot known as an airy disk. This spot typically exceeds 200 nanometer, far larger than the X-ray size of the emitting spot. If two emitting points are very close together, such as points P and Q shown here, their respective airy disks will overlap, making it impossible to resolve P and Q in the fluorescent image. The sizes of small molecules and biological macromolecules, as well as their distance between them, are typically below 200 nanometer. Subcellular structure also has a dimension below 200 nanometer. This situation leads to a challenge. Although we can locate this target using fluorescent probe, we cannot resolve their nano-sized structure due to the diffraction limit. In fact, without breaking the diffraction limit, we cannot be certain that the probe has targeted the correct molecule. To resolve nano-sized structure, it's necessary to break the diffraction limit first. After years of effort, scientists have come up with strategies and solutions. The first step is to actually identify the target molecule using fluorescent probes, such as A and B shown here. For this purpose, probes need to possess fluorogenicity, allowing for error-free recognition of the targets. Subsequently, the fluorescence on A and B is controlled to appear at different times. When point A emits light at time T1, the convolution of its point spread function yields the true position of A. Similarly, at time T2, the true position of B is all obtained. Once all points are localized, accumulating all points over time yields a superlution image of the structure. The process outlined here is the technical workflow of a single molecule localization techniques. Actually, all super-resolution imaging techniques adopt this time-for-space strategy. The key to implementing the time-for-space strategy lies in the ability to control when A and B points emit light. There are two main methods to achieve this. One approach is through fluorescence switches, which has led to the development of a super-resolution fluorescent microscope based on single molecular localization techniques, including PALM, STORM, PINT, and RESOLVE. Among these techniques, the fluorescence blinking controlled by the spiral ring switch of rhodamine is the most widely used. Another method is to control the excitation light with the spatial temporal resolution using structured light, such as SIM and STEP. However, these techniques still require fluorescence probe to first label the target, and then they require high brightness and photostability. Hence, rhodamine has become the the preferred flow for in supervisory imaging for its high brightness stability, and especially its derivative silica rhodanine. After gaining the ability to break the different limit, fluorescent probes first selectively recognize and light target molecule, and then fine structures are resolved through superlucent imaging. For example, what is shown here is superlucent imaging of the cell nucleus at the level of single molecular and nanometer scale resolution. Fluorescent probes must ensure specific molecular recognition without any error binding to non-target molecules, as this can lead to significant misinterpretation. Such probes are known as fluorogenic probes. In our work in recent years, we have permanently focused on improving spatial temporal resolution and dynamic superlucent imaging. By introducing intramolecular hydrogen bonds into rhodanine to control the switch of its spiral ring, we achieved continuous super resolution fluorescent imaging with a spatial resolution of 10 nanometer and a temporal resolution of 20 microseconds for up to 40 minutes. Through these studies, we gradually discovered that rhodanine can indeed break the different limit, but achieving true fluorogenic probe capability remains a challenge. So we go back to the difference of rhodanine, particularly the silicon rhodanine. Silicon rhodanine is the star in superlucent fluorescent imaging. This fluorophore was initially invented by Professor Xiaoyuan and Chen in 2008, and several years later, Professor Nagano modified its structure to give it brightness and darkness control properties. The most significant breakthrough came in 2013 when Professor Johnson discovered that silicon rhodanine aggregates in the form of a spiral ring. Upon interaction with the target, with the target protein, the spiral ring opens during this aggregation process, resulting in fluorescence image enhancement. Since then, silicon rhodanine has been widely used as a fluorogenic probe in super resolution imaging. And subsequently, Professor Lovitz discovered that the closed spiral ring structure formed by rhodanine aggregation contributes to the probe's cell membrane permeability. They also proposed using the ratio of open ring and closed ring structure to quantitatively evaluate the probe's fluorogenicity. As shown here, a higher ratio of open ring structures correlate with better selectivity in fluorescence labeling within cells. However, such fluorogenic probes can generate significant background interference. Our research several years ago have found that the spiral ring switch of silicon rhodanine is influenced by various external stimuli, including the aggregation, disaggregation process, external hydrogen bonding pace changes, and more. These changes in conditions can result not only produce rhodanine, not only produce fluorescence upon binding to target molecules, but also possibly producing fluorescence upon binding to non-targets, thus causing error fluorescence signals. At the nanoscale, these false signals can cause significant interference. For instance, silicon rhodanine simply stain mitochondria when entering cells. Here you can see the mitochondria imaging. But when there are target molecules in cells, we transfected hello protein to a nucleus here. At the low concentration of silicon rhodanine, there are hardly any noticeable false signals. However, when increasing the dye concentration besides the nucleus, you can see beside the nucleus also a significant amount of fluorescent signals can also observed on the mitochondria. As I mentioned before, with the increasing spatial resolution of supervisor imaging, for example, if we can down to 1 nanometer, there is a growing need for ideal fluorogenic probes that only emit light upon recognition of the target, only emit with the target. However, in reality, probes like silicon rhodamine struggle to activate this due to their environmental sensitivity, which lead to the significant background fluorescence interference. Besides the target, the silicon rhodamine also can emit light with 9 targets. The concept of a fluorogenic probe essentially involves in generating a gene-encoded fluorophore during the molecular recognition process, just like the biological principles of DNA, RNA to protein synthesis. Traditional fluorescent probes rely on the environmental sensitivity of the fluorophore. The traditional fluorescent probes contain the fluorophore, the linker, and the receptor. When the receptor selectively binds the target, and the molecular recognition will transfer the signal to the fluorophore and induce the fluorophore the fluorescent changes. And this is the fluorescent change is environment sensitive, but not just for the molecular recognition. So this kind of probe is not fluorogenic probe. If we want to get a fluorogenic probe, it's necessary to introduce gene-encoded components into the fluorescent probe that encode for fluorescent response, similarly to genetic encoding. Fluorescent proteins are genetically encoded. So here we introduce fluorescent protein into the silicon rhodamine probes. We insert fluorescent protein between the protein target and between protein target and the protein lab, the tag. After selective binding of the probes, the fluorescent protein is sighted, leading to fluorescence of particular rhodamine through FRET. However, those misplaced silicon rhodamine molecules lacking the fluorescent protein do not generate fluorescence FRET fluorescence. And these probes allow us to achieve genetically encoded fluorescence of organic fluorophores. Here, like the rhodamine, we give rhodamine the genetically encoded fluorescence, and we then design 2 sets. DFP with rhodamine, and another one is M12 with silicon rhodamine. We use 2 sets to demonstrate the generality of this strategy. And in the JFP, you can see in the JFP rhodamine group, it's evident that as the concentration of the fluorescent probe increase with increasing the concentration of probe, the left side is the excitation with the excitation of fluorescent protein. The right side is the rhodamine's excitation light. You can see the signal to noise ratio remains almost unchanged when it's using JFP excitation light. However, when using rhodamine's excitation light, the signal to noise ratio gradually decreased from 5 to decreased to 2.8. And also the similar phenomenon is also the same for M12 silicon rhodamine group. This indicates that by introducing fluorescent protein, the effective genetically fluorescence response of rhodamine in imaging has been achieved, truly making it a fluorogenic probe. And such a probe can be used to imagine different targets within cells, including proteins in cytoskeleton, nucleus, mitochondria, lysosomes, and etc. Due to the excellent photogenicity, these probes can be confidently used in multi-color imaging. The opening of the aggregated random spiral rings of interaction with the target molecules actually is due to the stability of the rhodamines ionic structure by nearby negative charges on the protein, because on the cation on the rhodamine, the fluorophore is the interaction to stabilize the cation to induce the ring opening reaction. Examples of commonly used proteins for this purpose include the snap tag and the halo tag. Snap tag and halo tag, they are all they contain some negative charges on the surface, interact with the rhodamine. But when the target proteins lack negative charge, that this aggregated rhodamine remain in the closed ring structure and doesn't emit fluorescence. This means that the rhodamine spiral ring switch reaction is not universal for designing photogenic probes. So we have to find another strategy to find to design the universal photogenic probes. SNAP tag is currently one of the most used protein tags, which attach to the SNAP protein by removing the guanine moiety from its substrate. We have found that the guanine readily interacts with the fluorophore, such as the nifedimine, shown here, and they can form further to form nanoparticles with the first quenching. Very good nanoparticles. And when these nanoparticles interact with the SNAP protein, they release the guanine from the quenching effect, leading to the significant difference in enhancement, 41-fold, and very quickly. Because guanine is involved in both protein recognition and fluorescence quenching, probes of this nature exhibit photogenic properties. Additionally, since the nifedimine fluorophore is neutral, different from the cationic rhodamine, and do not rely on the presence of the target protein, this type of probe is universally applicable photogenic probe in super resolution imaging. Then we use this probe. We achieve the long-term tracking of various cellular organelles, such as the cell membrane, mitochondria, the nucleus at the nanoscale. Based on this, we monitor the entire growth and the interaction process of the phallopodia within the race and the duration which is stayed from phallopodia budding to maturation, as well as the various formation modes of the phallopodia, such as fusion, evasion, and protrusion. We also monitor dynamic behaviors of mitochondria, revealing dynamic change in the mitochondria cristae, interaction between the mitochondria and the entire process of mitochondria autophagy. We also monitor the entire fusion process of a nuclear, revealing the time rate of a nuclear fusion. And here I would like to thank the Aggregate to give a chance to the public our work in Aggregate. So this is the conclusion and placebo slide. The application disaggregation fluorophore holds promise for designing multiple resolution fluorescent probes. However, the ultimate of the goal of resolution is to achieve spatial temporal resolution of down to 0.1 to 1 nanometer, milliseconds to microseconds spatial temporal resolution. And also multicolor imaging is a challenge. At least we need to have 9-color multicolor imaging at the same time. Then we need ideal photogenic probe in the future. And besides the photogenic property, these probes also need to have the smallest size, shortest label with the target, and also the brightness will be improved. Very big improvement to get the bright, maybe the brightest for in the future. And also the blinking properties, the key, also the key to achieve the very small, the spatial resolution down to 1.1 nanometer. Finally, I would like to thank my collaborators, my group members, my institute, and the funding support. The work on genetically encoded fluorescence was completed by Xiaoyuan and Miao Lu, and was work on generally photogenic probes was completed by Chen. And I'm happy to answer any questions for the audience. Thank you.
Thank you for the nice talk. Impressive. And I'm checking any questions. In the meantime, I'll ask one question. So far you used several core structure of the fluorophore for your purpose, which was previously used for the just confocal imaging. So what should be, or what can be the choice of your fluorophore for your super resolution imaging for this purpose? What is the first why you choose those fluorophores for your purpose?
Yeah, thank you. Thanks for the question. Good question. Actually, the first thing we considered to select the fluorophore for supervision imaging is the brightness and the photostability. Because for supervision imaging, they need the very bright. Because if the fluorophore is not bright, you cannot locate the position of the fluorophore. So the brightness is the first thing we should consider. And so the Rodman is the one was chosen by the scientists to use in supervision imaging. And also another thing is about the photostability. Because the light intensity is very high compared with the confocal, for instance, confocal microscope. The supervision, for example, the laser is very high, the intensity, laser intensity. So the fluorophore should be stable, even excited by the so high laser. So Rodman is also the photos. Maybe particularly for the silicon Rodman is is the most stable fluorophore until now. Cyanide dyes is also very bright, but the stability is not good. So as the very stage of the research, cyanide dye was chosen as the one for in super resolution imaging, but because its stability is not good, so after years later, Rodman was chosen instead of cyanide dye.
Okay, thanks. I have one more question from the audience. Metal nanocluster can be used for super resolution microscope?
Yeah, this is a good question, and sure, metal nanocluster can be used in super resolution microscope. these nanoclusters very small and very bright. But the thing is, it should be considered to if the nanocluster can emit light, and also the light emitting can be controlled to give a bright and dark switch, because only the bright dark switch can be used to break the diffraction limit. And also another thing should be considered is that the nanoconstruction should be, modified with organic molecules, because in super resolution imaging, the target is the single molecule. You should get some linkage, like use the click chemistry to bind with the target with only one bond. Then the nanoclusters or nanoparticles maybe is the problem for the the compound to introduce the ligand, only one ligand to find the target and have the only one bond between them.
Okay, if there is no further question, then due to the time limit, we can move to next speaker. So thanks again, Professor Xiaoyuan, for nice talk.
Thank you. Yeah, thank you.
Next speaker is Professor Mi-hee Lim at KAIST, and she got her bachelor and master at Ewha University, master with the Professor Won in our department, and she moved to MIT to obtain her PhD, and then postdoc at Caltech, and then he started her own independent work at the University of Michigan, Ann Arbor. Then he moved to UNIST 2013, and 2018 she joined the Department of Chemistry at KAIST. Currently, she is a KAIST endowed chair professor and the director of Center for the Metallo-Neuro-Proteinochemistry. Okay, and the title of her talk is Chemical Strategies to Study Multiple Facets in the Alzheimer's Disease. Please welcome her. Yeah.
Oh, thank you for nice introduction. Can you hear me very well?
Yes.
Yes, oh, thank you. So first of all, I'd like to thank you, the organizer, Professor Juyoung Yoon, for nice invitation. I'm very, very happy to have actually opportunity to present our work at this webinar. But the topic is little bit strange because I'm more bio-inorganic person. But let me share with you our chemical strategy to study multiple facets in Alzheimer's disease today. So since 2008, in our laboratory as a metallo-neuro-proteinochemistry group, in the community of the bio-inorganic chemistry and chemical biology, has been trying to elucidate the how metal-involved biological networks shown here are linked to healthy and disease state at atomic and molecular levels. We strongly believe that such a fundamental understanding allows us to develop the effective therapeutics and diagnostic for human disease. So among the human disease, our laboratory has been mainly focusing on investigating the pathology of the dementia, such including the Alzheimer's, vascular dementia, and other dementia as well shown here. So today I'd like to share with you our research outcome on the Alzheimer's disease. So as you know very well, Alzheimer's disease is a progressive neurodegenerative and fatal brain disease. More than 50 million people worldwide are suffering from Alzheimer's or related dementia. Alzheimer's is the most common form of dementia, but unfortunately, we don't know what is the actual cause of this disease would be. So recently, the US FDA has approved 2 new drugs, aducanumab and lecanemab. It's a human-derived antibody, but unfortunately, due to the side effects and the availability, the use of this drug has been very limited. So I would like to actually emphasize to you that there are no effective disease-modified drugs available for the Alzheimer's disease. So in the case of the Alzheimer's disease, you can simply recognize the shrinkage of the cortical and hippocampal area. In addition to that, when you're going to the inside a little bit, as a hallmark, the accumulation of the protein aggregate are shown here, such as the senile plaque mainly composed of the amyloid beta, A-beta peptide aggregate, as well as a neurofiber rectangle, mainly composed of the tau protein aggregates. So as you can see here, relatively speaking, upon progression of a disease, you can recognize the pathological features. But however, as I mentioned before, we don't know what is the actual cause of this disease. So many research group has been trying to, making significant effort on understanding the etiology of the Alzheimer's disease. For that effort, our laboratory is trying to get, obtaining fundamental understanding of the metal-involved biological network in Alzheimer's disease. So as shown here, we hypothesized that such a metal-centered network, and shown here, are very important for the development of the Alzheimer's disease. So far, our laboratory has been contributing, elucidating the interrelation among the metal and A-beta and ROS toward the toxicity of the Alzheimer's disease. And also, we have identified metal-bound A-beta species with metal A-beta complexes as an interconnector toward the toxicity triggered by these three factors. So how do we start such an investigation? So there are many hypotheses regarding the etiology of the Alzheimer's disease, but our laboratory has initially been interested in three of them: amyloid cascade, metal ion, and occipital stress. Based on our literature search at the time, when we actually studied this area, based on the literature, we can actually guess and also propose that metal A-beta complex can be the interconnector toward triggered by these three pathological factors. So let me briefly show you why our laboratory has thought metal A-beta complex could be the interconnector for these three factors toward the toxicity found in the Alzheimer's disease. So amyloid cascade hypothesis claimed that A-beta peptide are toxic species, A-beta peptide leading to the Alzheimer's disease. A-beta peptide are generated from the amyloid, the cleavage of the amyloid precursor protein by beta and gamma secretase. This generated a small peptide in monomeric species tend to aggregate, as shown here. So it goes to dimethylamine oligomer fibers. But recent study, many experimental evidence suggests that smaller aggregate species, including the dimethylamine oligomer, are more toxic than the fiber. But when you're looking at the inside of a plaque, then you can actually find a high concentration of the metal, such as zinc and copper and iron. And then you can see here the almost millimolar concentration of zinc and iron, 400 micromolar of the copper. So when this result, the data show, wow, it's many research groups, especially the bioinorganic research group, is trying to figure it out, direct interaction between the metal ion and A-beta species. So far, what has been known is a little bit well established. So copper and zinc can bind to any form of the A-beta species shown here. And you can see here, the metal binding is very reasonable on the biological constant in the system. In the case of the copper, is nanomolar concentration, and then 3-histidine and some of the antiminous residue are involved in the copper binding. So upon binding to the A-beta, they say copper can actually facilitate the A-beta obligation shown here and form and stabilize more potentially toxic A-beta oligomer. Not only for that, in the case of a redox active metal, in the copper case shown here, bound to the A-beta, in the presence of a dioxygen, electron, and proton, they can generate the reactive oxygen species. So if I summarize for you what I have been talking to you, this metal ion can directly bound to the A-beta, and then they can influence the A-beta aggregation and conformation. Not only for that, in the case of a redox active metal, can bound to the A-beta under biological condition, especially in the presence of dioxygen, they can generate the ROS catalytically and then leading to the occipital stress. So based on what I have been actually presented to you, you can already, maybe a little bit convinced metal A-beta complex can be the interconnector toward the toxicity observed by these three factors. But when we actually study, this is totally based on the literature. So we have been actually curious, what is actually direct linked between the reactivity of the Metal-Abeta complexes and development of the Alzheimer's disease. So it has been proposed, but we didn't actually found the experimental evidence then. So we want to first answer this aspect. But when we actually, started this research area, we immediately figured it out. We didn't actually have the right chemical tool or method to investigate the Metal-Abeta complexes in vitro and in vivo, especially in the brain of the Alzheimer's disease. So we stepped back and then we developed some small molecule based on our rational structure-based design, and the small molecule as a chemical tool, they can target the Metal-Abeta complexes and control their reactivity. So reactivity of the Metal-Abeta complexes include the Metal-Abeta aggregation, generating more toxic oligomeric species, as well as the Metal-Abeta mediated ROS formation, leading to the oxidative stress. So we have been actually designed for the various small molecule, and then we found the very interesting reactivity, towards the Metal-Abeta complexes. But among them, one of the very simple compound, you can see here, the bidentate ligand, the simple bidentate ligand, very two-step synthesis, very simple. And this compound, this amine compound, we call it L2B, is the winner because this compound is relatively soluble in water, and also relatively stable under metabolic condition. And this compound is very specific for the Metal-Abeta species. Using the L2B, we have been able to control the Metal-Abeta aggregation, as well as over the metal-free A-beta aggregation, as well as we can reduce the ROS production by copper A-beta species. Not only for that, this L2B was able to go into the brain of the Alzheimer's disease transgenic mice, and then we can actually was able to lower the amount of the Metal-Abeta species. And then we found, consequently, we show that the cognitive improvement of the cognitive defect. So this compound, even though it's just a simple the metal chelator, this mode of action was not the metal chelator. Mode of action was the ternary complex formation with the Metal-Abeta complexes. And then you can see here, if you generate a ternary complex like this, then you can interact with the A-beta. So we can control the A-beta aggregation. Not only for that, this compound can interact with the metal center. So we can control the geometry, so not to actually generate the ROS as well. So using the L2B, we have been able to report experimental evidence that Metal-Abeta complexes could be directly involved in the pathology of the Alzheimer's disease. So from this study, we have been actually proposed Metal-Abeta suggests Metal-Abeta complex as a target for the drug discovery. But when we actually studied the Metal-Abeta complexes, one of my graduate students now, she's the professor in the University of Seoul, Juyoung Han, was wondering which one is actually more toxic, copper A-beta and zinc A-beta. But if you consider the reactivities, then aggregation and ROS formation, obviously copper A-beta was the more toxic. But when you're actually looking at this copper A-beta space, how can we actually control the copper A-beta specifically? But at that time, we didn't actually find any chemical strategy. So she was actually looking at into the more mechanistic approach. So what she was thinking, this was the copper A-beta monomeric complexes. You can see here 2-histidine and some N-terminus residue was involved. So she was thinking, can we actually directly modify the metal binding site? Then maybe we can control the copper coordination as well as the A-beta aggregation. So she developed some small molecule that can directly, chemically modify the coordination sphere of the copper A-beta complexes. So we have been able to, chemically modify. Such a modification allow us to change the copper coordination as well as the A-beta aggregation. So the compound was not that complicated molecule. The compound is just like looking like this, very similar to L2B, but we put it at the thiophene because it's the worst metal binding site. That is actually key because that is the transiently generated some of the ternary complex with the copper A-beta, but that is on and off. So that open side of coordination side can be binded to O2. So we are going to utilize the O2 chemistry decision here. Let me show you. So in the L1 case, in the presence of the copper A beta, under aerobic condition, this L1 is actually through the ND-alkylation generate aldehyde amine. This aldehyde, one of the histidine residue, and actually I want to point it out, that histidine 14. A beta actually has 3 histidine, 6, 13, and 14. And this is specifically the 14 histidine residue that covalently bind, interact with this aldehyde and making the covalent bond, and also this copper A beta in the presence of the L1, of course with the O2, they can actually generate the copper O2 intermediate that can actually oxidize the histidine residue. So generate the 2-oxohistidine. Such an activity can be also, actually we can actually achieve is because of this L1, redox potential of this L1 is very similar to the copper 1 and copper 2 A beta, the redox potential of copper 1, copper 2 A beta complexes. So they can communicate the electron transfer. So, because of the time, I'm going to immediately show you our mechanism, how this compound works. So in the copper A beta with L1, transiently generated ternary complexes shown here, and then electron transfer between the ligand and copper. So one electron is from the ligand to the metal center. So ligand is oxidized, and then copper is reduced, generated the copper 2 to copper 1. And the copper 1 is can do the beautiful copper O2 chemistry. So copper O2 chemistry, and through ND-alkylation, our compound is generated, the cleavage by generate the producing the aldehyde and amine, and then one of the histidine residue is the covalent to the nuclear attack with the aldehyde and the covalent bond formation. Not only for that, another copper O2 chemistry, copper O2, these transient complexes that can generate the multiple oxygen species, the reactive oxygen species, and then copper O2 intermediate, especially not the free, the ROS, but that is actually can oxidize the histidine into the 2-oxohistidine. So such the reactivity of the L1 case can occur through direct the interaction between our ligand and A beta followed by the copper O2 chemistry. So you don't need to add something. So you have the pathological the factor copper A beta species, just add in the L1, and then you are just utilizing the O2. It's already available in biological system. And then I want to point it out that this dual modification is by L1 compound is very specific for the copper A beta species. So such a chemical modification, as we expected, the aggregation pathway of the copper A beta was specifically controlled, not for the other compound or our control compound, because we tuning the redox potential, and the metal-free case doesn't work, and then the toxicity has been improved because of that, the modification. So Professor Ji-Hyun Han was able to find a very small molecule that can specifically modify the coordination sphere of the copper A beta complexes, and then they can actually allow us to change the copper coordination to A beta, and then the aggregation and toxicity of the resultant, the modified the A beta species. So when you actually study for the metal A beta complexes, we have been realized immediately there are many, many interrelated pathological factors exist. Upon progression in the brain of the Alzheimer's disease, so upon progression of a disease. So even though we're looking at the 3 of the hypotheses, then we can see that here metal ion, metal-free A beta, ROS, and the metal A beta complex. From this hypothesis, now we have to include this one from our study. These 4 pathological factors, exist, and the form, and then they are actually there. And then they can communicate with each other. So we have been thinking, then maybe can we actually design for some small molecule? We can actually target all of the factors shown here and then control their reactivity. So using the one in the compound. So basically what we need a novel multifunctional molecule. So our targets are metal and metal-free A beta, metal A beta, and ROS. So we know that what kind of structural moiety to achieve such an interaction, each of the pathological factor, and then what kind of property we need for the biological application. So we basically need the structure, all of the required structure moiety for the desired property, has to be in the one single structure entity. So we have been looking at the natural product, and the natural product usually they have the very poor biological applicable property, such as the BBB blood-brain barrier permeability. So we just structurally optimize and improve their property, and not only for this effort, we actually freshly from the scratch we can actually design for such a multifunctional molecule. But when we actually designed for this molecule, we found that a very small molecule, very compact molecule. We don't need a very complicated structure molecule. Basically, this dimethyl phenylene diamine compound is shown here. It's actually maybe what we are looking at the reactivity we can actually carry on. So look at here. So even though the structure of this DMPD is very simple, but the DMPD structure include the structure required for A-beta interaction, metal interaction, metal-A-beta interaction. Not only for that, DMPD is already known to be antioxidant and then relatively soluble in water. And based on our in vitro, in vivo study suggests that this DMPD can going into the brain. So, but it's very, very small, simple molecule. But using this compound, we have been able to really redirect both metal-free and metal-bound A-beta peptide into the upper pathway and unstructured A-beta aggregate. And these unstructured A-beta aggregate are reported to be the less toxic or non-toxic. And then obviously antioxidant property is carried on, so ROS can be controlled. So our multiple pathological factor can be controlled by the DMPD. So this one, maybe you are going to wonder, it's very small, how this one has such a reactivities. So we did the very, various mechanism study, and in the presence and absence of the metal ions. And then what we found that this DMPD is actually intramolecular cross-linked between this compound, but the transformed compound and A-beta. So we have when we have this DMPD in solution, just one electron oxidant and then radical species is the form. This is a beautiful purple color, and then this one is relatively stable. But in the presence of the A-beta under pathological condition, in both the absence and presence of the metal ion, this DMPD is chemically transformed to the benzoquinone, and then primary amine from the lysine residue and N-terminus priamine can generate this covalent bond adduct. Such a covalent bond adduct allow us to change completely this aggregation pathway, fiberization into the upper pathway, generating unstructured A-beta species, and that suggests to be non-toxic. And this compound is the specific reactivity is not only one. This DMPD was going into the brain and the Alzheimer's disease transgenic mice. We observed the significant reduction of the amyloid load and cognitive, and subsequently we can improve the cognitive function as well. So even though very tiny, but we have a very specific interaction with the A-beta, and then we have the very nice reactivity toward the pathology, the amyloid pathology, against the amyloid pathology. The key was because this DMPD was redox active. And then of course the minimum structure is right there for the interaction with the pathological factor. So after we found the DMPD, my graduate student Mingun Kim, and he actually was immediately make the library of such the compact compound, because he found the very minimalist principle for making such a small molecules. So you basically need a basic structure like this, and then if you put it the substituent, obviously we can tune the redox potential, and then whatever you want to target the pathological factor, then the sum of the redox potential, then you will be able to tune into your compound. Then, if you have the right potential communication, redox potential is right there, then in vitro and in vivo efficacy was on. So, it's very simple, small, the required minimum structure, and then you change some redox potential chemical property, then you will be able to achieve such a reactivity as well. So we generated a various small molecule based on this principle. So So in 2008, we have been trying to generate some small molecule to elucidate some pathogenesis of the Alzheimer's disease and contribute to the new discoveries. But since 2022, I have been very fortunate to actually achieve, receive some of the funding. So we have been actually moving forward to discovering the new biomarker or drug target in Alzheimer's disease. But our new discovery is also from the fundamental understanding of the matter involved the biological network is shown here. So in the brain, maybe you already know there are already the labile, the metal ion and static metal ion, and the weaker binding and tighter binding. The metal is right there. And then also we have a neuropeptide, responsible for the neuronal and hormone and the signaling. And also we have a neuron-related, the protein that's related to neurodegeneration. And also the neurodegenerative disease, we observe the formation and the formation of amyloidogenic protein and accumulation of their aggregates. So all, as you can see shown here, is all, player is the metal and protein and neuropeptide and neuro-related protein, amyloid protein is right there. So we have been thinking maybe they're, from the understanding of their direct interaction, maybe we can find some toxic species or non-toxic species. And then maybe we can actually find the new biomarker or therapeutic target for this Alzheimer's disease state. So, as a proof of concept, our laboratory recently found a neuropeptide. This is called somatostatin. They can interact with the pathological factor associated with Alzheimer's disease. This SST can bind the metal, such as in the particularly copper case, through the N-terminus and then the C-terminus. And then the binding is not the important. In the binding is not the only case. Upon binding the copper, what is happening? This conformation change. Such a conformation change triggered the aggregation of somatostatin. This aggregation phenomenon, it actually blocked the binding to the receptor. So supposed to be SST has to bind to the receptor and then transmit the signaling, but because of the aggregation, they lost the normal function. However, but in the presence of the metal and A-beta species, and the pathological condition related to Alzheimer's disease, they generate a ternary complexes. Such a ternary complex formation actually can, control, modulate, reduce, control the aggregation and toxicity of both metal-free and the metal-bound A-beta species. So actually, SST has the new function, the new function under pathological condition. So we found that under pathological condition, this is neuropeptide can actually lose the normal function, but it's actually gained the new function. So our study suggests that metal SST and the metal SST and A-beta complexes can be the new biomarker or therapeutic target in Alzheimer's disease. In addition to the SST, we have very recently found a new peptide as the intramolecular promoter for the aggregation and toxicity of the metal-free and metal-bound A-beta species associated with the Alzheimer's disease. That peptide is this C-terminal fragment of the amyloid precursor protein. Inside the cell, this C-terminal fragment is generated by the caspase-mediated cleavage. This C-terminal cleavage, the protein C31 peptide is actually, we first time actually, reported a direct interaction between the metal A-beta. From all of the biological method we confirm, but not only for this direct interaction. So because of the direct interaction, if we're looking at our MD simulation data, the self-recognition side, very critical for the aggregation of A-beta. This part is the secondary structure is changed to actually facilitate the formation of the beta sheet. So that means, this A-beta because of this interaction, A-beta aggregation can be facilitated. So as you expected from our interaction research, we actually confirmed that in vitro, if you're looking at here, in the presence of the cis-Shaoyuan peptide, the aggregation of A-beta has been facilitated in both metal-free and the metal-bound species, metal-containing condition. You can see here clearly, even the shorter the incubation, in the presence of APP-cis-Shaoyuan, we see the significant, increased amount of the A-beta fibers. And that the toxicity was increased, as we expected. Not only for because of this is inside the cell layer, so we actually directly incorporated injected our two peptide A-beta and cis-Shaoyuan into the cell through the micro injection. We observed that the increased amount of the A-beta aggregate in the presence of the APP-cis-Shaoyuan. Not only for that, we incorporated, injected directly two peptide in the hippocampus area in the brain of the mice brain, and then after the several days, we observed the more aggregate species as well as we observed the more inflammation. So we can find that. This aggregation was facilitated. Of course, the toxicity was increased in vitro, in living cell, as well as in vivo. So through this study, we suggest maybe controlling the formation of cis-Shaoyuan, as well as the interaction between the cis-Shaoyuan A-beta would be the new therapeutic target for Alzheimer's disease. Okay, so I would like to finish my presentation today. I would like to thank you to my current member and past member, and then I have the fantastic collaborator in Korea and outside, and then very nice supporting from the funding agency. And then thank you so much for your attention. And if you have any question, I would love to answer.
Thank you, Professor Lim, for the fascinating talk. It was great. And let me start the question. If the pharoptosis or chiroptosis occurs, with what's the concern or effect for the A-beta aggregation, or is there any chance for those mechanisms?
So I think the pharoptosis and then today this is iron related, and also the other one is cupritis. This is copper related. This is all metal, the metal. Then it's actually the aggregation, the A-beta aggregation and toxicity we observed is the more increasing. So in another way to this area is actually controlling and regulating that phenomenon. Then it's A-beta species, A-beta related amyloid pathology has been improved. So the metal and A-beta is directly, that study and some of the recent following study, current published paper and then the result actually really suggests that this metal and A-beta communication is very critical for the pathology in Alzheimer's disease. Yeah.
Okay, thank you. Actually, the floor is open for the question. If you have any question, you can actually ask to the speaker right now. Okay, if there is no further question, I think we can conclude our webinar. Since Professor Shawn Chen could not make it due to his unexpected flight delay, so we feel sorry for that. But we had very nice 3 talks from Professor Jiang Ping Qi and Zhao Qi and Professor Mi-Hee Lim. Thank you so much for nice presentations to the speakers. And also, thank you so much for the attendance to the audience. Thank you again. And if Ben does not have any concluding remark,
Yeah,
do you
no
have something
further.
to say?
Yeah.
Okay.
Thank you very much, Professor Yoon, for the wonderful webinar you have organized. And I hope we will see each other very soon somewhere.
Okay. Thank you so much for everyone. Thank you. We'll see you again in the near future. Thank you. Bye-bye.
Thank you.
Thank you.
Bye-bye.
Bye-bye.
Thank you. Bye.
Bye.